Hello, and a very warm welcome to this Vaccine Insights webinar on process and analytical insights for GMP manufacturing of mRNA-Lipid nanoparticles. I'm Charlotte Barker, editor of Vaccine Insights, and joining me today we have a stellar panel who will discuss challenges and opportunities for GMP manufacturing for mRNA-LNPs, including process, analytics, data management, and facility considerations. As always, do feel free to pose a question to our panelists at any time throughout the event using the Ask a Question box, and we'll try to get to it during the session. There'll be 2 polls today as well, so please look out for those on your screen. We'd really love to hear your feedback. Okay, so let's introduce the panel. So first up, we have Emmanuelle Cameau, who's the Gene Therapy Strategic Technology Partnership Leader, EMEA, at Pall Corporation. Emmanuelle has more than 15 years of experience in biotechnology process development and GMP production, and she's highly skilled in the field of cell culture applications. Our next panelist, Shell Ip, is the Client Learning and Scientific Content Manager at Precision NanoSystems. He's been serving in technical communication and training roles in Precision NanoSystems' commercial team since 2015. He combines his multidisciplinary background with an aptitude for communication to convey complex concepts to diverse audiences through various media. Next up is Linda Mathiasson, who's Strategic Customer Leader in the Nucleic Acid Therapeutics Business Unit at Cytiva. She's responsible for technical and scientific engagement and collaborations with customers in the global RNA application community. Linda has more than 22 years of experience in the life sciences industry. Our next panelist is Katarina Stenklo, who is an enterprise commercial activation and end-to-end solutions leader at Cytiva. Katarina is an experienced biopharma professional with over 19 years of industry experience, and she has extensive experience in developing end-to-end manufacturing solutions, including process workflows, process equipment, automation, consumables, and facilities. Our final panelist is Peiqing Zhang, who's the Strategic Technology Partnership Leader for Gene Therapy at Pall Corporation, responsible for technical engagement with customers and the wider gene therapy community across the Asia-Pacific. Peiqing has more than 15 years of experience in biotech research, development, and commercial manufacturing. So, without further ado, let's get the panel discussion started. Okay, brilliant. If our speakers can make sure that you're unmuted and have your camera turned on, that'd be great. Wonderful. So, first question for the panel: what are the advantages and disadvantages of viral vectors, lipid nanoparticles, and other gene delivery technologies? How can one best navigate the options and choose which to use? Emmanuelle, I'll come to you first.
Thanks, Charlotte. So I think it's a bit difficult to answer that question straightforward, because it's not really black and white. Because the stages of where these modalities are is different. The industry has been developing and improving viral vector manufacturing since several years now, and we are now at a point where viral vectors allow for tissue targeting, for example, or organ targeting, and industry is also even looking at engineering capsids to target even further and also reduce the immune response. On the other side, viral vector technologies are more expensive to manufacture as the cost of those, and also the process is longer compared to, for example, LNPs. So I think depending on what is the goal of the therapy, what is the target tissue, whether there is a modality that works, or if several works, then it needs to be balanced between what are the different advantages of the different ones that are available for the indication. But I think in the future this will evolve. I think today there's not really a guide, as referring to the question on how to choose for one or the other. But I tend to believe that once we know a bit more, and once these modalities, and especially the new ones around LNPs, mRNA, also we see a lot of exosomes developing, are more mature, my thought is that they will each have a predicted role. Let's say like in a toolbox, they will each have a preferred mode of action, mode of administration, whether it is local, whether it is in the blood. Like that's what I see from where I stand.
Absolutely. And Shell, do you have anything to add there?
Sure. Thanks, Charlotte. Yeah, absolutely. What Emmanuelle articulated is really important. Just the ability to, or the wealth of options here for drug developers is really great for being able to reach these different target product profiles, even conceive of different target profiles, and to be able to attain those in some way. And I think that benefit extends beyond drug developers as well. It's really helpful for patients and users as well. So one great example is with the rollout of the COVID-19 vaccines, there was some difficulty outside of the US and continental Europe to get supply of these mRNA vaccines when they first rolled out. So many of us had to rely on viral vector vaccines, and that's certainly the case in Canada and very likely elsewhere in the world as well. So I think the more variety that there is, the more choice there is, and ultimately this both of those things working and coexisting will be really beneficial for the industry as a whole.
Definitely. Moving on to our next question. As mRNA therapeutics move towards personalized medicine, what process control considerations need to be taken into account as the scale decreases compared with pandemic responses? I was going to come to Linda here, but I think she's having a few technical difficulties. She's not been able to join us yet. Hopefully she'll be able to later. But so I'll come to Katarina for this one.
Yes, so I think in principle, it is the same requirements for process control as in the larger scales, but the spans, it's actually from one patient and back to the same patient. So actually the control starts much earlier, and the regulatory requirements are the same regardless of the size of the batch. So you can say that you will have to apply the same regulatory requirement, but also the GMP requirements and the controls of traceability will start already from going from the patients, and to really make sure that you're getting back to the same one. And also, as you're going towards personalized, the scales goes, of course, down, but still so how much down, it will depend on the therapy, because how many doses and and then also the size of the dose. But still, from an analytical perspective, you're overproducing just to cover the analytics. So that's one aspect of it. And another difference is that we might actually need to do a little bit more work on the upside of actually where you're doing prediction modeling on how will these work for these specific, when you're going towards personalized. So you're doing more modeling on to get the right effect on side effects and so on. So that's so I think that will be important coming, and also the manufacturability. How do you really manufacture it in these smaller scales? So that's where I would see how it moves.
Brilliant, thank you. And Peiqing, I'll come to you next.
Yeah, thanks, Charlotte. So I fully agree with what Katarina shared. So as we scale down from a pandemic vaccine, using mRNA-LNP as a novel modality to more personalized side of medicine, right? So maybe, I think it's a term subject to debate over here, whether we call it personalized medicine or at a higher level, precision medicine or stratified medicine. I think regardless, as Katarina pointed, for sure it's going to involve smaller batch sizes and more production runs for the same facility that they would actually otherwise use for pandemic vaccines. So I think that's going to bring a lot of changes to the supply chain, to the manufacturing cadence. For example, from conventional stockpiling model to the kind of make-to-order business model on more personalized or stratified medicine side of things. So I think fundamentally it actually requires, even stronger than before, a higher degree of manufacturing flexibility on the drug manufacturer side, being it MAH or CDMO entities. So I think, from a process design point of view, it's going to involve, more than before, the use of single-use technologies, standardized manufacturing operations, and how to couple those standardized unit operations together to allow the drug manufacturer to quickly switch from one product to another. But specific to process control, I think there are two aspects of the requirement, in my opinion. One is actually a more platform approach for manufacturing execution. So that, again, that would actually mean less change to the process for the drug manufacturer as they change product to product. And also, as Katarina mentioned, there's also analytical challenge to make sure that, it's also going to be a short period of time for the drug developer to develop the analytical protocol to validate it, and perhaps using a bracket approach to validate platform or standard method in order to cover different personalized medicine. So in my opinion, those are the challenges, and also perhaps some food for thoughts when we move to the personalized space.
Thank you, Peiqing. And in light of all that, how can technology providers quickly adapt to support those changing industry needs? Katarina, we'll come back to you for this one. Katarina, I think you're on mute. Katarina, you're on mute, I'm afraid. We can't hear you. Can you try and unmute yourself?
Yeah, no, the unmuting button disappeared.
Oh,
sorry.
Sorry about that.
So,
do you want me to repeat the question?
Yes, please do.
Okay.
Sorry.
How can technology providers quickly adapt to support the changing needs of industry?
Yes, so as I said, we see that it will change quite quickly, and actually what we can do is to make sure that we have a standardized platform that can be utilized for a lot of different, are prepared to handle different projects, because as we said earlier, there will be a lot of multi-products in the facility. So having really standardized but flexible manufacturing is what we can have here. So the same equipment can be scaled both up and down. If you have a single-use flow path, that can be switched and have a very short changeover between the different products, but also have a possibility to have the unit ops being as plug and play inside the workflow. So if you need it for one process, you plug it in, but if you don't need it for the other, you can plug it out. So just design with flexibility, and that goes with both the platform but also the facilities. We are thinking about manufacturability from the beginning and really invest in something that is flexible that can adapt with whatever changes there will be in the future.
Wonderful. And Emmanuelle?
Yeah, I agree. I would add to that the platform approach also, and the fact that using the same equipment and consumables and therefore materials at small and at large scale through the different phases also has an impact on the regulatory side, because then you have really the same contact parts, the same like a smaller scale validation could be translated into larger scale, or at least could help win time at that point. So and it's also really important to through this approach really link to the technology provider on getting the right guidance, for example, on the different validation steps. For example, for sterile filtration filters, that this is really important to manage and to assess from the very early step of the process.
Absolutely. And moving on, thinking about the analytical challenges. Well, first off, what are the analytical challenges in mRNA-LNP testing and characterization? Peiqing?
I think if we talk specifically on the analytical challenges itself can be a topic for many hours, but maybe just at a very high level, I think in my opinion, there are two aspects of analytical challenges when we relate to mRNA-LNP. In my opinion, the first aspect of the challenge is really coming from mRNA-LNP being a novel drug modality. So the resulting challenge is really a rather limited knowledge space or knowledge base as compared to what we know about other conventional biologics, for example, monoclonal antibodies or recombinant proteins. And the second aspect, which is basically related to the first one, is the still developing, rapid developing analytical tools and technologies to ascertain, the safety and efficacy of mRNA-LNP. So let me briefly touch upon the first aspects. So I think the unique characteristics of mRNA-LNP as a modality as compared to conventional modality like protein-based drugs, right, is if you look at it, let's say the drug substance being the free mRNA and the drug product is actually the complex formulation being lipid nanoparticles. So definitely you can see you actually need two comprehensive sets, which are partially overlapping sets of analytical technologies to take care of the in-process testing, release testing, stability testing, and also extended characterization. One set for the drug substance, another set for the drug product, right? And also, as I mentioned, this is rather a novel modality, so our knowledge space is really limited. So I think to analytical scientists, and also I'm here making a reference to the draft version of ICHQ 14, which is the analytical procedure development. It is rather a challenge to clearly define what is the analytical target profile for the analytical scientists to start select the assay to initially define the ranges of the assay parameters in order to ascertain, the drug is actually made to the acceptable quality or specification limits. So I think that's the number one challenge. So more specifically, we can talk about, for example, specific to mRNA drug substance, the 5' capping, identity, the relative abundance of it, the 3' poly A tail, the length, the variance, and related to that, and also some product-related impurities like double-stranded RNAs, shortmers, longmers, modified nucleotides, right? All these, I think later perhaps Shell could also chip in to talk about the whole, new set of challenges brought about by the LNP part as well. And on the second aspect, in terms of the analytical technologies, I think there's, to begin with, there's always a strong leveraging on the traditional modalities. So for example, the use of liquid chromatography, capillary electrophoresis to ascertain, the drug purity. So I think that's okay. However, there's still, as we know, room for improvement to, gain further resolution, detection sensitivity for those assays. And specifically for mRNA-LNP, I'll just would like to mention, the increasing, expanding use of mass spectrometry in coupled manner, like CEMS or LCMS, for not just, identification, but also for absolute quantitation of the drug purity and impurity, conversely. So I think taken together, those two are what I see as the challenges and also the opportunities for analyzing mRNA-LNP products.
And thank you. And Katarina, would you agree with those?
Yes, I would actually. This is really, I think you got really good answers there from Peiqing. So
wonderful. And anything then, Shell, did you want to add something? I know Peiqing mentioned that you might have something to add there.
Yeah, sure. So I'm not the analytical expert at Precision Nano. We've got Adam Crow and his team here who've worked pretty closely with folks at SciEx to help develop some of these techniques. And recently had a webinar about detecting these lipid adducts. So this is a very specific issue for LNPs. So the ionizable lipids can degrade into these N oxides, which then covalently bind to RNA specifically to produce these adducts. So being able to quantify those, and that's a fairly specific product that you need to be able to detect for this particular drug product. So I think developing these techniques and as they advance will bring us closer to being able to quantify this. The other thing that Peiqing mentioned was, what are the specific analytical methodologies for the drug product itself? I think if you're coming from a biologics or a small molecule space, you may not immediately appreciate that how much the size of the particle can affect the safety and function. So a particle size really affects where the particles end up in the body following injection and depending on the route of injection. But there have been many studies showing that where the particles end up in the body, their ultimate fate is highly correlated with particle size. So being able to determine particle size, make sure that that is the particles in that population are monodispersed. And typically we use dynamic light scattering for this. It's the challenges there is that they have to sample a relatively large quantity of particles. So you take a relatively small sample, but within that sample, there are a lot of particles and variation can be quite high. And compare that to sort of more fine-grained techniques where you're looking at almost individual particles, for example, with electron microscopy. The latter gives you much more detail about the individual particles, but is not statistically very relevant when you're looking at hundreds of particles when your sample consists of billions. So those are the sort of the challenges. And I think one advantage is that at least dynamic light scattering is quite fast and pretty amenable to in-process testing as well. So there's some groups, for example, Yvonne Perry's group in Strathclyde have published a paper where they've integrated at-line testing of using dynamic light scattering for continuous process manufacturing for nanoparticles.
Absolutely. Thank you, Shell. And what are some of the challenges in scaling mRNA and LNPs during the drug substance process and formulated bulk process? I know Linda's more the expert on drug substance. She's unfortunately still not been able to join us, but we hope to have her on soon. But Katarina, do you have anything you could comment there on the drug substance side?
Yeah, no, generally you need to think about where you're going to be at the end, because a lot of time what we're seeing now, it's an early sort of concept where you're working on, right? A lot of things is happening on the lab bench, early stages, but what you're doing there might not be possible in the manufacturing scale. So actually think about where do I want to be? Make sure that you have scalable methods, because that will save you time so you don't have to go back afterwards and work your method. And also sometimes you need to know what the facility can do, where you want due to the end process, and also the manufacturing equipment. But also beware that there might be steps that you cannot just scale up. You might have to scale out sometimes. So that's where you need to consider. You need to, consider where you want to be. Think manufacturing from the beginning is what I should say, and choose the right technology, because some of the choices that you are making in very early can have a huge effect in the end for the batch cost and the setup. So that's where you need to model and think manufacturing already when you're at the early development stages.
Absolutely. And Shell, do you want to talk a bit more on the drug product side of things?
Yeah, sure. Yeah, I can speak probably more specifically to the nanoparticle aspect here, but it's important to be able to use the same or similar technology that you're using at the lab bench that you're going to attempt to use at the larger scales. And I think that's, to Katarina's point, being able to think ahead to what you're going to do in the clinic and essentially keep that in mind as you're doing the development phase. And some of the challenges at the, for the mixing of the LNPs, the whole process involves mixing an aqueous phase with an organic phase to basically trigger the self-assembly. And the control of that mixing is going to be very important in order to have very consistent results. So what you would like, and sort of to my point I was making about the analytics, the particle size is so crucial to the function and biological fate of the drug product that it becomes incumbent on us to really consider the ability to manufacture these in a very controlled way. And so being able to, how do you control the assembly of millions of molecules into a particle? It happens kind of spontaneously. You don't really think a lot about what's going on at the molecular level, but having that understanding and being able to ensure that all of the materials that flow through your process undergo the same conditions, and that what you're doing at the bench scale is the same, essentially the same process that you're doing at the clinical scale is going to be important.
Brilliant, thank you, Shell. Emmanuelle, anything to add?
I think my colleagues covered most, but I mean, going back to what Katarina was saying, which is exactly accurate. I mean, it's exactly the same for any therapy, whether it's a viral vector, it's the same concept. We always tell the customers, you need to think manufacturing. But for the mRNA-LNP, some specific examples are, for example, I mean, I think a lot of customers come to us and they're making mRNA from a kit. So very early, you need to switch from the kit to reagents and a protocol that is with the same thing, same starting materials that you would use in a medium, large scale, like as Katarina was saying in the final process. Because if you delay that, then you have a risk that the final product is not really what you were working on, and then you get maybe delays. So it's really important to have that focus, do that gymnastic, to think ahead, as Katarina was saying, on every step from the very early stage. Same for the purification of the mRNA. I mean, when we look at kind of the platform we usually promote in our end-to-end solutions, this is based with very scalable technologies such as chromatography, TFF, modules, filtration. But if you choose something a bit more exotic that is not scalable until a bit longer in the process, maybe already in clinical, then you will have to change at some point, and that will delay. So it's really important to have that focus early stage.
Absolutely. And how can digitalization improve mRNA-LNP manufacturing and increase efficiency? Katarina, I gather you've worked on some of the GMP manufacturing side of that.
Yeah, no, so there could be two aspects of this one. First, you can, as we mentioned earlier, there are some modeling that you can do. Actually, you can do some steps in silico, especially looking at the DNA sequence and prediction of the mRNA and optimizing actually the manufacturing in silico. And the other aspect is digitalization aspect is on the manufacturing side. How will you move away from the pen and paper records and actually having an electronic system for executing a full manufacturing and then doing batch release and so on for the final product? Because that takes time, and considering that you can do a lot more batches per year, with mRNA, it's a short process. You don't have weeks of cell culture. You will have the potential of having a lot of batches, but that is a lot of papers, a lot of things that is going to be released. So that is somewhere where we can start adapting much earlier on the digitalization journey.
Definitely. And Peiqing?
Yeah, I think I agree with Katarina. She covered it very well. I would just share or echo. So in my opinion, digitalization opportunities come in three areas when it comes to mRNA-LNP. So we are actually now talking about finally a cell-free enzymatic process for the synthesis part, and for the purification is actually highly similar at a high level, similar technologies that's applied for conventional biologics. So as Katarina mentioned, which I fully agree, there's definitely a very appealing opportunities for process modeling, because now the whole reaction is actually simpler. There's no black box being the cell factory, right? But there's enzymatic homogeneous solution involved, standard purification unit operation that's involved. So I'll say definitely opportunity for process modeling. here I would like to extend a little bit. I think in recent years, we hear a lot about the promise of digital twinning. So to me, that actually sounds another opportunity for us to further develop and apply to mRNA-LNP to use digital tools to gain the foresight about your process or manufacturing execution. How is it going to be, right? Before it's actually being tested in a QC lab standalone. Another area is really on the analytics. I do hope there will be opportunities for us to finally extend the use of process analytical technologies in a digital manner to, at line, if not, fully online, inform the manufacturing execution, how it's going to be, and, to also accelerate batch release and gain the understanding of the stability profile ahead of time. The third aspect, I think Katarina also mentioned, is really the opportunity to digitalize your manufacturing execution to make your manufacturing shop floor more nimble and potentially more compliant, because you are actually automating a lot of things independent of the operators. So those are the three areas where I see the opportunities for digitalization for this model.
Absolutely. And what about the cost? How can the cost of mRNA production be lowered? Shell, do you want to take this one?
Sure. Yeah. So I think Peiqing mentioned cell-free synthesis of the RNA, and I think that there is still a cell-based portion of this, which is the production of the DNA template. So I think going down the route of a cell-free DNA template synthesis could help to reduce costs. I think naturally, as these technologies mature, we're going to see a reduction in cost. But ultimately, the RNA is going to be the primary cost driver in this equation. And one of the routes of reducing the cost is actually to reduce the necessary dose. And this is where technologies like self-amplifying RNA come into place. So these are relatively new technologies, but they promise to really reduce the necessary dose by orders of magnitude. So if you can imagine, instead of like a 30 microgram dose, having a 1 microgram per dose, you can reduce the cost significantly, especially because the RNA is that cost driver.
Absolutely. And I'm going to come back to you again for the next question, Shell. How can one leverage partnerships with solution providers to accelerate those drug development timelines?
Yeah, I think trying to find a solution provider that can provide an end-to-end solution will really help reduce those timelines. So for drug developers having to deal with a variety of different vendors and trying to coordinate materials being sent from one place to another, or to source these materials from different vendors and ensuring their quality can be quite challenging. And I think being able to consolidate that as much as possible, to have a solution where your RNA manufacturing can be integrated with the drug product formulation will ultimately streamline things for a lot of clients.
Absolutely. I was going to come to Katarina next, but I think we may have briefly lost her. She might have been eaten by that dog.
No, I'm here.
Wonderful.
I'm here. Yeah.
Wonderful. Did you have any thoughts on how you can leverage partnerships with solution providers to accelerate drug development timelines?
Yeah, so even if we are not solution providers, we are not producing drugs itself, but we are the experts on our equipment and our systems. So actually, for developing them, we have a lot of knowledge on how to do things. So actually partnering with a solution provider, we can also support in process development and also make sure that you get access to all that expertise that we have inside our company. And also, there is actually an opportunity to get training where we have our sites to get training on the equipment that you are going to use further on. So that can speed up your actually release of your product or getting up and running for manufacturing. So I think that's another aspect.
Absolutely. Okay, next question we've got here. What applications for mRNA technology are you most excited about for the future? Nice question. Katarina, we'll come back to you on this one.
Oh, I'm actually, so I think that I'm really well, excited for the whole field because it's happening so much, and actually being able to meet that. You have seen the Cephi's lab to jab in 100 days. To have that aspect on actually going to solve something that is emerging very quickly, can we be even quicker than we was for the pandemic, but also going into these more personalized cancer therapies and seeing. I think that's where I'm seeing how as a really exciting future here.
Absolutely. Shell, do you have some thoughts on this? What are you excited about?
Yeah, thank you, Charlotte. Cancer therapy does have a personal spot in my heart for me. I think a lot of the technologies from which the RNA vaccines for COVID emerged, came from work where companies were developing cancer vaccines. So I think a lot of that was put aside to focus intensely on COVID. And I think now that we have vaccines on the market for that, the shift can be back to these cancer medicines and personalized medicine. So I think that's a really, at least selfishly, what I'd like to see happen. But if I were to try to be more objective, I think I'm really just excited for the opportunities that exist there. The technology is very powerful. So you can not only express a gene of interest, like in the case of mRNA, but the same technology can be used to regulate other genes through gene silencing, delivery of CRISPR-Cas9 components as well through RNA components is also possible. And you have groups like Intellia Therapeutics who have one candidate in clinical trials at the moment. So I'm really just excited for what possibilities exist. Maybe even 10 years ago, I remember going to conferences and trying to convince people that mRNA is the way to go. And look how far we've come in just a few years. So I shudder to think, what's possible that we haven't at this point yet conceived of. So that's exciting to me.
Wonderful. 100% agree. Did anyone have any to add? Anything they're excited about they want to talk about? I think Shell's covered us all. Wonderful. Okay, so compared with conventional biologics facilities, what special considerations need to be taken into account when designing mRNA-LNP facilities? Katarina, let's come to you with this.
Yeah, so I would say that normally the mRNA manufacturing doesn't require any high biosafety level, so it's still biosafety level 1. But considering that mRNA is really sensitive to RNases and degradation, you should have dedicated space. Don't mix with any other products. You should have a really good, of course, airlock going in, so you do a changeover and with clothes and masks, etc., because the human is the worst enemy for mRNA. And but if we are looking at the LNP formation, you need to consider that you will handle solvents. So if you are going to do large batches or actually multiple batches, but where you in the end will have a lot of solvents moving through the facility, there might be areas there you actually need to rate. It might not have to be the manufacturing suite for LNPs. It could be the storage next to it where you make up your lipids and so on. So so that's something to consider. Again, think about where you want to be when you are manufacturing at full scale. So you really prepare your facility to handle that, and be aware that the regulations around this one is different in different parts of the world. So you need to check with your local authorities here to make sure that you're getting the correct rating of the building.
Absolutely. And Peiqing?
Yeah, so Katarina is an expert in facility design, so she covered it well. Technically, I just want to echo, I think more specifically for mRNA manufacturing, especially on the drug substance side of things, one particular challenge is really the RNase control, right? So this is basically ubiquitously present and known to be, degrading our precious drug substance. So in this case, this has to be carefully thought about in terms of the design of your flow path, as trivial as that, right? And the other thing which Kat mentioned is really, for example, the ATEX rating requirement if you are handling a large volume of organic solvent when it comes to the formulation side of things. One thing I would like to connect back to the previous discussion as we touched upon personalized medicine side of things. So I think for a drug manufacturer, especially true for platform companies like CDMOs, I think it's important to bear in mind, as Kat mentioned, where you want to be. I think chances are the drug manufacturer want to be on multiple drugs, right? So I think that's the challenge. It's actually the very wide range of skills that you need to consider when you design a facility. So we are talking about a very large volume, especially when it comes to post-LNP formulation side of applications versus a very small volume when we talk about personalized medicine. So I think that order of magnitude difference has to be carefully thought about and designed into your processes and implemented into your manufacturing execution. So it's really the scalability challenge in designing the facility is what I would like to add on.
Brilliant. Thank you, Peiqing. And what are some of the opportunities and challenges then as lipid nanoparticles expand to other applications, such as CAR T-cell therapy? Shell, what are your thoughts on that?
Sure. Thank you, Charlotte. I think with CAR T therapies, gene editing is a very important tool in this toolkit. And I think one of the real opportunities for LNPs is that they are quite amenable to multiple treatments on the same population of cells. So this is really good for example, if you wanted to create an allogeneic cell therapy, you can first use gene editing components to essentially knock out endogenous receptors and to make basically a universal donor cell, and then treat the same population a second time in order to introduce exogenous genes to express your CAR. So I think that's one real advantage of LNPs potentially in this space. And we actually have an off-the-shelf LNP kit you can just buy off the shelf and start experimenting with this in human T cells. I'll add also that this is sort of ex vivo gene therapy and engineered cell therapies. I think with LNPs, there are folks at least in the preclinical phases, exploring in vivo gene editing for these types of engineered cell therapies.
Absolutely. Emmanuelle?
Yeah, I mean, nothing much to add other than, yeah, I mean, technically, these LNPs can, bring in genetic material, whether it is DNA, mRNA, to any cell, whether it is out of the body or in the body. So I think this certainly opens up a very large amount of potential utilizations and applications. And it will be really, really interesting to see how these are developed towards in the next years, and how that maybe makes the actual industry evolve on all these modalities. So I think it's an amazing new tool we have, and I'm really curious to see what it will bring.
Absolutely. Katarina, anything to add there?
No, I think both Emmanuelle and Shell had covered it. So thank you.
Wonderful. Okay, so another question here. What are the unique challenges of manufacturing mRNA-based products? Shell, let's come back to you on this.
Yeah, I think for me, it's just the acknowledgement that the formulation plays quite a large role in the safety and efficacy of the drug product, perhaps more so than we're used to with other biologics. So the lipids themselves are not sort of passive excipients in the drug product. So they play much more of an active role in being able to sort of regulate or understand the quality aspects that affect outcomes there, but also to be able to control that formulation step and to give that the due consideration that's necessary to ensure quality of these drug products.
Absolutely. Katarina?
Yes, no, if you look at the more of the upstream of the formulation, of course, it is to avoid making double-stranded RNA, for instance. So that is one of the challenges, to really do the design and the IVT reaction correctly. And then one of the challenges is that on the purification side is that this really happened very quickly. We were not having so much specifically products designed specifically for mRNA and mRNA purification. So, and this is where I'm really excited on how the future will look like, because I know that everyone is working on that side to make better products that is fit for mRNA when it comes to capacity, the way of actually getting a much more optimized workflow all through. And again, right now, some of the techniques that has been used is really been used on the lab bench and not really fit for scaling up. So that's where some of the challenges is. But I hope we will see a future with the new products that is really designed towards mRNA.
Absolutely. Okay, so another question here. Audience member asks, what microfiltration, ultrafiltration, and other separation techniques, such as cross-flow, are used in production? Are there steps from monoclonal antibody production, downstream, upstream, etc., that are not used? Emmanuelle, do you want to take that one?
Yeah, sure. So as I quickly mentioned earlier, once the IVT is done, the techniques to purify the mRNA from the rest of the IVT reagents, are like common use techniques in bioprocessing that come actually from the mAb industry at first, right? So it's classically filtration, could be depth filtration, sterile filtration, TFF, whether it is with cassettes, with hollow fibers, and chromatography. This is optional. Some manufacturers choose to not have chromatography steps in their process. We find that it's more optimized, it's better to have at least one chromatography step, maybe two, depending on the purity that is expected. But there are many different ways of making mRNA, and yes, they use this classical method.
Wonderful. And another question here, how much process development time, both for lipid nanoparticles and for AAV, does a flexible platform save customers versus non-platform approaches? I don't know someone want to volunteer for that one?
I can take a stab at it. So I think that, well, for mRNA, there is no really a fully platformed process right now, because what we are seeing, it really varies much on this for each mRNA. You need to do some process optimization. But having an equipment platform, on the other hand, will save you time that you know how to work it out. You have a good starting point for your process, and that is how you can save time. And also knowing where you would be, as we have been talking about, know where you want to be in the end. So again, you don't get away from optimizing, but knowing what to use and how to start will save you time.
Absolutely.
Yeah, perhaps I'll add that I think even though we can't really answer this, very precisely in terms of exactly how much time one might save, but the whole platform approach is really one of the advantages of the RNA-LNP technology as a whole, because minor changes in, say, the sequence of your RNA, you can use that to tailor it to express nearly any protein. But from a physical-chemical perspective, that RNA molecule is very similar. And that allows you to have very modular approaches to developing a variety of drugs. And the whole platform process will allow you to really facilitate multi-product facilities. So, one facility that can really manufacture more than one drug product, And that's really the main advantage. And I think those two things can't really be decoupled.
Yeah. I would like to quickly add, as solution providers or suppliers, we are not in a very good position to inform the audience in terms of absolute number of months. What is the time saving that you can achieve? Over here, I would like to just borrow the experience, I think industry-wise, that we are aware from the monoclonal antibody world, right? So we're talking about, I think in a few years ago, I think 20 months development time cycle from getting the DNA sequence to put it to IND filing. That's kind of that was the industry norm. However, in recent years, I think you are talking about 12 months being really not uncommon, right? A lot of companies can achieve taking the DNA sequence in 12 months, they can deliver it to IND filing, right? And also in a few isolated cases, when it comes to infectious disease, we have also seen example of single-digit number of months from DNA to IND. So that's actually the benefit of using platform technologies, process sciences, analytical tools to accelerate your development cycle to make sure it actually meets the quality requirement to initiate phase 1 trial. Again, we are not in a position to inform what does that time saving mean when it comes to mRNA-LNP, but the same concept would apply, I would argue.
Brilliant, thank you. And how do you see the adoption and implementation of process analytical tools, for example, LNP particle characterization, for the future, and the use of inline PAT tools for such analysis in future GMP manufacturing for these technologies? Where do you see the gaps today, and where do you see that going in future? I don't know who'd like to jump in here.
I can maybe try to take a stab at this. That I think some of the gaps are that trying to understand what in-process analytical techniques you can use that are quick enough to be able to provide information that you can then feed back into your process, and how developing that relationship between these characteristics and the process controls that you have at your fingertips. And I think understanding that feedback loop and what to look out for, what you can change to alter it, and how quickly you can feed that back is something that still needs a lot of development work in the LNP space. I think just because there is quite a bit of variation depending on your actual materials, your actual formulation, the process controls and what parameter space that you have available for you will change as you change those materials. And I think as the technology evolves and the field matures, we'll have a better understanding to be able to make broader associations between those things and be able to do that kind of in-process control a bit better.
Absolutely.
I would like to quickly add on top of Shell's comment, very well covered. Would like to extend, I think this is actually a very good question. I would like to extend it a little bit to think, in addition to inline or online monitoring or PAT tools, we could also consider at-line, right? Anything that can accelerate the analytical turnaround to inform the manufacturing execution and also to assure the quality, right? So at-line monitoring, for example, the use of aseptic sampling to take the sample and put it to a otherwise standalone, but in this case, coupled analytical tools like, who knows, maybe CMS or LCMS, right? For rapid analysis in a kind of automated fashion. So I think that could be also a possible workaround to implement process analytical technologies in the broader term. In terms of the gaps, I won't say the gap. I think this is always the wish as an industry player we want to accomplish together, working together, across the value chain, right? Between suppliers, drug developers, manufacturers, and also regulatory bodies, right? To make sure that we clearly define, as Shell mentioned, what is actually the goal of implementing PAT, right? And what is the validation strategy? What are the critical control points?
Sorry there, Peiqing, you just muted for the last sentence there. Sorry about that. A little technical problem.
Yeah, so sorry about it. So I was just saying that it actually takes the whole industry approach, involving suppliers like us, the drug developers, manufacturers, and also the regulatory agencies to align on what is actually the benefit of implementing process analytical technologies, whether inline or outline. What's the benefit? What's the validation strategy? What's the control strategy to ensure that it's actually fit for purpose and bring the benefits at the end of the day to assure safety and efficacy to the patients?
Absolutely, that's a great point. And what are the main challenges in downstream processing at the industrial scale for mRNA production, especially thinking about tangential flow filtration in the presence of thick buffers, for example, containing cryoprotectants?
Yeah, I can maybe start there. So TFF has been used for complicated technologies, complicated products since a long time. So and also delicate products. So there is a broad offering, and there is, I mean, as technology providers, we know how to operate the system so that there are no issues. So of course, if you have something very thick, this might have an impact on the molecular cutoff you might use, or whether you use a suspended screen, for example, in a cassette, or if you use a hollow fiber on the diameter of the hollow fiber, and of course on the process of parameters, process conditions you work. But this doesn't impact the scalability. So whatever you define at small scale that works at small scale has no reason not to be scalable, provided you are able to control that your product and the state it is stays the same through the process and doesn't get altered through being in a larger volume. So that is always to be controlled. And referring back to the analytical, that's where the in-process controls are really, really important. And then, but one of the big challenges of the downstream at that point, I think it's more, or the scale up, it's more around the LNP creation. And that is what are at PNI, and Shell can comment, it's really the expertise. And we know how to scale that process to the scale that is needed without any alteration.
Yeah, thanks, Emmanuelle. I'll maybe then add to that. We're constantly developing some of those downstream processes to adapt them for LNPs. So as Emmanuelle mentioned, these technologies have been around for a while and have been used in a lot of other biologics applications. But it turns out the application of it towards LNPs are still a bit of an area where a lot of development still needs to be done. There isn't a lot of guidance if you look through the literature about what parameters are important and what range of parameters that you should focus on for LNPs. And ultimately, what we've come to learn is that depending on the specifics of your actual formulation, those can change, and there's not one set of parameters that's going to work for everyone. You have to sort of optimize it on a case-by-case basis. But once you do have it optimized at a relatively small scale, then those should translate pretty easily to larger scales, which is really the advantage there. But the one thing to consider about LNPs is that depending on how long your process is, you need to consider what is the stability of your drug product in those conditions for that period of time. So, storage of LNPs for vaccines and distribution are, as we know at the moment, minus 80. During the processing, though, a lot of this processing has to occur at room temperature, and you need to be able to understand the stability of that product at room temperature for however long you need to do the downstream. And that's in addition to some of the challenges that were mentioned in the question with the viscosity and other issues.
Brilliant. Well, thank you all very much for answering those questions. That is all we've got time for today, I'm afraid. Any audience questions that we didn't get to, we're going to be trying to reply to those by email. So hopefully you won't be left unanswered. The webinar will be available on demand tomorrow, so look out for an email from us with the link. And I think all that's left is to thank Emmanuelle, Shell, Katarina and Peiqing once more for joining us today. And thank you to the audience for listening. We hope you join us again soon.